This is a cell. It is currently in interphase. Look, it's saying hello. Now this cell has just gone through mitosis and is starting fresh in the G1 phase. During this phase, it will grow and replicate all the parts it needs to be a fully functioning cell. Later, it will enter the S phase. This is when the cell synthesizes a lot of DNA, which in its current form is known as chromatin. After this, the cell will enter the second gap phase, or G2 phase. During this time, chromosomes start to form from the chromatin. Enzymes and structural components are synthesized, and the centrioles move to the opposite poles of the cell. This is the end of interphase, and the cell is ready to begin mitosis. But before we get to that, let's talk about why mitosis is important. If a cell never divided, an organism would never grow or repair damaged cells. Now let's continue by zooming in on the nucleus and the chromatin. The first phase of mitosis is prophase. Throughout this period, chromatin continues to condense to form paired chromosomes. Also, the nucleolus disappears and the nuclear membrane begins to break down. The next phase is metaphase. During this time, spindle fibers formed by the centrioles attach to the centromere of the chromosome, which have lined up at the metaphase plate or the center of the cell. During anaphase, the sister chromatids split as they are pulled apart by the spindle fibers. This causes the chromatids to move to opposite poles of the cell. Next is telophase. This is when the cell membrane pinches inwards and forms the cleavage furrow. Finally, there is cytokinesis, when the cell membrane splits the cytoplasm and the organelle duplicates into two new daughter cells. These new cells then return to interphase and the cycle repeats. Now we must leave these two cells and return to the macroscopic level.